Endospore staining. This is I believe would be the fourth staining method you are going to learn today. And gram staining, one staining method, and direct indirect staining, second staining method you learned. And this would be endospore staining. And by reading the slides, you are capable of knowing the concept of vegetative cells and endospore cells. If you get a question, please go to your TA office hour to ask. Then the ideal image we want to got under our microscope would be like this: endospore formed inside of our vegetative cells. And we are going to use saffron to stain vegetative cells in pink color, so you all know saffron is in pink color. And then we are going to use the bright and green dye to stain endospore cells. And we are going to use Bacillus series to stain get our stain slides. And for this result, we have an online handout which will be due this week, Saturday midnight. Remember to turn in your online handout. If you pass the due day, that's fine. That's 3 points. And any students missed the due day of online handout, and so your TA will ignore such emails if you pass the due dates of your online handout. So remember, after you finish watching the video, please go to online handout to answer these 2 questions. The procedure, basically what I am going to do, I'll get an empty tube and add 10 drops of distilled water. After 10 drops of distilled water, I'm going to get a slant. What is slant? I'm going to show you.
Here.
So slant is basically agar, a neutron agar inside, but inside of test tube, and you basically get a surface. So this is slant. Bacteria grow on the surface of the slant, and I'm going to use my inoculating loop, scrape off some cells from the slant, and mix with my 10 drops of distilled water. And then I'm going to add 5 drops of bright and green dye, mix with my endospore or not Bacillus series. Then I'm going to put into boiling temperature, boiling water for 10 minutes. Again, this is a harsh condition you artificially created to have endospore formation. Then we are going to, after you treat it for 10 minutes, take out, and I'm going to prepare the slides. I basically use my inoculating loop, and onto my slides, get some cells onto my slides, air dry. Air dry is especially important for your direct staining method, and you heat fix it for 1 minute over the incinerator. Then stain with the saffron for 2 minutes. So saffron stain your vegetative cells. So basically this experiment, you use 2 different dyes to do your experiment. One, you use bright and green dye to stain your endospore cells. Another, you use saffron to stain your vegetative cells. Then we are going to view under microscope. In the slides we introduced to you, 10 drops. 1, 2, 3, 4, several drops of, I think should be enough. 10 drops of distilled water, and then you are going to get inoculating loop, sterilize it, and for 30 seconds, take it out, let it cool down. After it cool down, I'm going to cool down. I touch here, and then I am going to get some bacterial cells. I'm going to go closer a little bit, let you to see how I scrape some bacterial cells off from the slant like this. I get some bacterial cells. Probably I need to get more. And then I go into mix with my 10 drops of distilled water. After that, I need to sterilize my inoculating loop again. And I want to vortex it. There is a machine we could vortex our test tube. Some students think it's really funny to do so, like this. Here is our bright and green dye. What I am going to do, 5 drops. 1, 2, 3, 4, 5. Okay, very dense green colored. Go to our 100 Celsius boiling water for 10 minutes. 10 minutes done, I use my grabber, safely take it out, otherwise it will be very hot for me. So I let it cool a little bit and perform the staining step. So everyone, I finished taking out my 10 minutes treatment. Basically, this treatment allows the bright and green dye drive into endospore, and I'm going to get some loop of the cells onto my slides, and then I'm going to allow a little bit of time to air dry my samples. After it air dry, basically I'm going to heat fix it for 1 minute. After I heat fix it, I'm going to use sulfurine to stain it, and later I'm going to show you the 100 times total magnification of our endospore staining slides.